to be met. Today we are going to talk about the glycolysis, which is the first step of the carbohydrate metabolism. So in this video we are going to talk about the carbohydrate metabolism, which is composed of main three steps. First one is glycolysis, second one is Krebs cycle, and the third one is electron transport chain. So let's continue our lesson with the first step. Although the word glycolysis is complex in appearance, it can be easily broken down into two parts as glyco and lysis. Glyco means glucose and the lysis means breakdown. So now we are going to learn about the pathway of glucose breakdown. Before going to the deep content, let's talk about basic but most important facts about glycolysis. So the first fact is, this is the first step of the main metabolic pathways of cellular respiration. And the second fact is, it occurs in the cytoplasm of most prokaryotic and all eukaryotic cells. It means this can be seen as a common process in both eukaryotic and prokaryotic cells. Third fact is, glycolysis is the metabolic pathway that breaks down glucose into a few other products. We will discuss this in details in a few more minutes. So, our fourth fact is, it is an anaerobic process, which means none of its 10 steps involve the use of oxygen. So, it occurs in both aerobic and anaerobic states. So the next fact is, as you can see, it starts with one molecule of glucose and produces two pyruvate molecules. If not, at the end we can have two pyruvate molecules. Next one is, ATP is initially required for this procedure to be occurred. It means energy is consumed during first and third steps. Let's jump to a quick overview of this glycolysis pathway. It will help you to realize what happens, what are the phases, what will be produced at the end of this process. Likewise, you will be able to gain a basic idea of this entire process. So, glycolysis can be divided into two main phases. During the first phase, glucose molecule is converted into glyceraldehyde 3-phosphate molecule and energy is consumed. During the second phase, that glyceraldehyde 3-phosphate molecule is converted into a pyruvate molecule and energy is released. So let's see what happens during this step 1. So step 1 is defined as the phosphorylation of glucose. It means glucose is phosphorylated and form glucose 6-phosphate. So during this process, glucose molecule is converted into glucose 6-phosphate molecule and one ATP molecule is converted into one ADP molecule. It means energy is consumed. So this reaction is catalyzed by the enzyme called hexokinase. But do you know what is the meaning of the word phosphorylation? Well, let's talk about it. So, you can see here is a glucose molecule and you already know it is composed of 6 carbon atoms since it is a hexose sugar. I ask you to suppose this yellow color spherical chain represents those 6 carbon atoms. So, that glucose molecule is converting into a glucose 6-phosphate molecule. When you look at that name, you should know that it contains a phosphate group. Of course, yes, during this phosphorylation, one group of phosphate binds to the 6th carbon atom of the glucose molecule. So, that's why we call that molecule as glucose 6-phosphate. So, phosphorylation is a biochemical process that involves the addition of phosphate to an organic compound derived from ATP. So, my important tip number 1 is, if there is phosphorylation, it means ATP molecule should be converted into a ADP molecule because glucose gains energy by being phosphorylated at the expense of one ATP. Most important fact is, this reaction is irreversible. So let's see what is an irreversible reaction. Irreversible reactions can convert reactants into products, but products cannot convert back to reactants. I think my tip number 2 will help you to remember this fact. So remember, you can easily make a cake from its ingredients, but can you go the same process in backwards? I mean, you can't turn a cake into its ingredients again, right? Well, now we know what happens in the step 1. So let's move on to the step 2. In the second step, glucose 6-phosphate molecule is converted into fructose 6-phosphate molecule. But when you look at these two structures, you will see both the structures are similar. So it's because of the isomerization. What is isomerization? Isomerization is a process in which a molecule, ionomolecular fragment is transformed into an isomer with a different chemical structure. So in here, one aldose molecule is converted into a ketose molecule. So this reaction is irreversible and phosphohexoisomerase enzyme is catalyze this reaction. So, my tip number 3 will help you to remember this enzyme. Before jumping to the full name, let's talk about the second part of the name of this enzyme. It is isomerase, right? So now you know what is the meaning of that word. It defines the process of isomerization that I explained in the second step, right? So, my tip is, if the word isomerase is used at the end of an enzyme found in this process, it defines that enzyme has catalyzed a reaction related to isomerization. Now you know how this second part was created. So let's talk about the first part. So did you know we can also use the word glucose phosphate isomerase instead of phosphohexoisomerase? Because this enzyme plays the role of isomerizing the glucose 6-phosphate molecule. So let's remember it as glucose phosphate isomerase. Well, step 3 is defined as the phosphorylation of fructose 6-phosphate. According to that, again a phosphorylation should be occurred, right? So let's see what's going on. Fructose 6-phosphate molecule is converted into fructose 1,6-bisphosphate molecule. Well, can you detect any difference between these two molecules? As you can see, there are two phosphate groups are attached with both first and sixth carbon atoms of the fructose 1,6-bisphosphate molecule, right? That's why we call this molecule as fructose 1,6-bisphosphate. Well, what's the meaning of the word bis? We use the word bis to define if there are two identical but separated complex groups in one molecule. So, now we know if there is a phosphorylation, certainly one ATP molecule should be converted into an ADP molecule. It means energy is consumed. This reaction is irreversible and the phosphofructokinase enzyme is catalyzed this reaction. Now let's see how to remember this enzyme. In the step 1 we found an enzyme called hexokinase, and in the step 3 we found an enzyme called phosphofructokinase, right? So, can you detect any similarity between these two enzymes? Well, the part kinase is common to both of them. So let's see what's the meaning of that word. Kinase is a type of enzyme that adds phosphate to other molecules, if not, catalyze the transfer of phosphate groups from a high energy phosphate containing molecules such as ATP. Well, the fourth step is defined as the cleavage of fructose 1,6-bisphosphate molecule, because during this step, fructose 1,6-bisphosphate molecule is split into two different molecules. First one is dihydroxyacetone phosphate molecule and the second one is glyceraldehyde 3-phosphate molecule. Let's move on to the fifth step. Actually, these compounds are isomers. So, during the fifth step, these two molecules can be interconverted into each other because of this isomerization process. This isomerization reaction is catalyzed by the triphosphate isomerase enzyme. So, tip number 7 is this glyceraldehyde 3-phosphate molecule is being utilized during this glycolysis pathway. So, the equilibrium of this reaction is always toward this glyceraldehyde 3-phosphate molecule. So, this concludes the several steps in the first phase of the glycolysis pathway. You should keep in mind energy is consumed during this entire phase. Let's see what a reversible reaction is. Well, a reversible reaction is a reaction which the conversion of reactants to products and the conversion of products to reactants occur at the same time. In these type of reactions, reactants can be converted into products and products also can be converted into reactants at the same time. I will explain it with a simple example. So, remember this example as my tip number 4. Well, you can get ice by freezing liquid water, and as well as you can get liquid water by melting that ice again. So that's why we call these type of reactions as reversible reactions. In the sixth step, glyceraldehyde 3-phosphate molecule is converted into 1,3-bisphosphate molecule. So, as you can see, this molecule is composed of two phosphate groups. Most important fact is, during this reaction, one molecule of NAD is reduced to one molecule of NADH. So, tip number 8 is, every reaction of the second phase of glycolysis happens twice and produces twice. Therefore, in here, two glyceraldehyde molecules are converted into two molecules of 1,3-bisphosphoglycerate. And since 2 moles of glyceraldehyde 3-phosphates are formed from 1 mole of glucose, 2 NADH molecules are generated. This reaction is irreversible and glyceraldehyde 3-phosphate dehydrogenase enzyme is catalyzed this reaction. It was the overview of the sixth step. Let's clarify it further. Glyceraldehyde 3-phosphate dehydrogenase enzyme is catalyze the reduction of oxidation. So, as you can see, this is the glyceraldehyde 3-phosphate molecule which is composed of 1 phosphate group and 2 hydrogens. During this reaction, this enzyme removes the hydrogen atom from the first and second carbons of glyceraldehyde 3-phosphate molecule and then transfers them to NAD to form NADH. Important fact is, one water molecule in the cytoplasm is also involved with this reaction. After removing hydrogen atoms from the first and second carbon of the glyceraldehyde 3-phosphate molecule, one OH group is get attached with the first carbon and create an intermediate molecule which is very unstable. And then it get attached with another phosphate group and become more stable, and that molecule is called as 1,3-bisphosphoglycerate. So this is how glyceraldehyde 3-phosphate molecule becomes 1,3-bisphosphoglycerate molecule. So during the 7th step, that 1,3-bisphosphoglycerate molecules are converted into 2 molecules of 3-phosphoglycerate. So as you can see, this new molecule is composed of only one phosphate group. Let's see how was it happen. Well, we call this process as a substrate level phosphorylation. It means the high energy bond between the first carbon atom and the phosphate group of 1,3-bisphosphoglycerate molecule is broken down during this conversion. That various energy is used to convert 2 ATP molecules into 2 ATP molecules, and this is the first energy producing step of this glycolysis pathway, and this reaction is irreversible. So during the 8th step, 3-phosphoglycerate molecules are converted into 2 molecules of phosphoglycerate. So the only difference between these 2 molecules is the position of the phosphate group. During this conversion, phosphate group has changed the position from the 3rd carbon atom to the 2nd carbon atom. So energy is not produced or consumed. This reaction is catalyzed by the phosphoglycerate mutase enzyme, and this reaction is also irreversible. Let's move on to the 9th step. During this step, 2 phosphoglycerate molecules are converted into 2 molecules of PEP, if not phosphoenol pyruvate. So this reaction is catalyzed by the enolase enzyme. It removes a molecule of water from 2 phosphoglycerate to form PEP. Well, let's move on to the last step. In this step, 2 phosphoenol pyruvate molecules are converted into 2 pyruvate molecules. This step is defined as transfer of phosphate from phosphoenol pyruvate because the enzyme pyruvate kinase transfers a phosphate group from PEP to ADP. Therefore, 2 ADP molecules are converted into 2 ATP molecules. Finally, you have to know the net amount of the ATP gained at the end of this glycolysis pathway. So let's calculate. Well, 2 ATP molecules are produced at the end of the 7th step, and another 2 ATP molecules are produced at the end of the 10th step. So totally 4 ATP molecules are produced at the end of this glycolysis pathway. But you should remember there were energy consumed step as well. So 2 ATP were consumed during the 1st and 3rd steps. Therefore, the net amount of ATP can be calculated as 2 ATPs. Let's see what are the end results of this glycolysis pathway. Well, one glucose molecule is converted into 2 pyruvate molecules, and during this conversion, 2 ATP molecules and 2 NADH molecules are produced. Great! That is the end of the glycolysis. But what will happen to that pyruvate molecule? It enters the Krebs cycle, if not citric acid cycle. We will talk about it in our next video. Stay tuned!